I want you to just imagine the plot of basically every single pandemic disaster movie you've ever seen. You know the exact scene, right?
Oh, yeah. The classic opening sequence.
Exactly. The camera slowly pans into this dark, remote cave, or maybe like a super dense jungle canopy. And there's this virus. It's silently circulating in a bat or a monkey. And over time, it starts mutating.
Like it's hitting the gym.
Yeah. It's essentially going through this cinematic training montage, just, leveling up its genetic abilities in the shadows, unlocking new traits one by one until it is perfectly, terrifyingly evolved to attack humanity. I mean, it's a fantastic Hollywood narrative, but what if we told you that story is completely wrong?
Yeah, it's definitely a compelling visual, but it heavily distorts how zoonosis actually works.
And by zoonosis, we mean...
Right, sorry. The process of a pathogen jumping from an animal into a human, the movie version, really skews how that happens in reality.
Yeah.
And correcting that narrative isn't just about, ruining movie trivia. It fundamentally changes how we monitor the globe and prepare for the next massive health crisis.
Spot on. So today our mission for this deep dive into the source material is to explore this really groundbreaking 2026 study. It was published in the journal Cell by Havens, Kosakowski-Pond, Wertheim, and their colleagues.
It's a massive paper.
It really is. We are basically going out into the field today to decode the evolutionary history of the world's most notorious zoonotic viruses. We're talking Ebola, SARS-CoV-2, Marburg, Mpox, and even, and this is my favorite part, this mysterious ghost strain of the 1977 flu.
That part is fascinating, yeah.
Right. So we are hunting for the answer to one massive question. Do viruses actually need to adapt before they can start a human pandemic?
And to really set the stakes here for everyone listening, we have to look at the two competing frameworks for how these outbreaks happen.
Okay, lay them out for us.
So the traditional view is exactly your movie plot. It posits that a virus must first undergo this period of intense adaptation. It has to spend time either in its original animal reservoir or, in some intermediate bridging animal. It's basically picking the locks to unlock the specific molecular keys required to infect humans.
Right, like it needs a warm-up period to figure us out.
Precisely. But the alternative view suggests a reality that is, it's much more random. And frankly, a bit more unnerving.
Unnerving is a good word for it?
Yeah, because it proposes that the virus already has the keys. It's already fully capable of infecting us and spreading. It doesn't need to train at all. It just needs a lucky break, like a physical opportunity to cross the species barrier.
So in that framework, the species barrier is just a passive physical sieve, not an active biological training ground.
Exactly. It's just about contact, not evolution.
Okay, let's unpack this, because how do you actually figure out which theory is right? I mean, we can't go back in time and sit in a bat cave with a notepad watching a virus mutate. To figure this out, you have to read the virus's evolutionary diary. But a mutation is just a mutation, right? Like,
Sure.
How do you know if a virus is changing because it absolutely has to in order to survive, or just because RNA is inherently messy and just makes mistakes?
That is the core challenge. And to solve it, you use a field called phylogenetics. Basically, you build this massive family tree of the viruses.
Yeah.
But more importantly, you analyze the specific types of mutations that are accumulating along the branches of that tree.
Because not all genetic changes are created equal.
Exactly. They really aren't.
Right. So let's use a visual memory hook here for you guys listening to lock this in. Imagine a virus's genetic code is a massive manuscript for a novel. When the virus replicates, it's making copies of this manuscript, but it's a super sloppy typist, so it makes mistakes.
Lots of mistakes.
Lots of them. Now, some of these mistakes are what geneticists call synonymous mutations. Think of these as minor typos, maybe a misplaced comma or, spelling the word color with a U. The reader doesn't even notice, and the fundamental meaning of the sentence doesn't change one bit. The physical protein that the gene makes stays exactly the same.
Yes, and the mechanism behind that, just to get into the weeds for a second, is the redundancy built into our genetic code. Multiple different 3-letter RNA sequences, which we call codons, can actually produce the exact same amino acid.
Oh, wow. So you can swap a letter and still get the exact same building block.
Precisely. So synonymous mutations, the DS mutations, don't change the virus's physical structure at all. And because the physical structure doesn't change, these mutations are functionally invisible to natural selection.
They just kind of fly under the radar.
Right. They just accumulate steadily over time, basically like dust settling in a room.
Okay, but then you have the big ones, right? The non-synonymous mutations. In our manuscript analogy, this is not a typo. This is the author crossing out an entire paragraph and completely rewriting a character's arc, or just changing the ending of the book.
It's a structural change.
Yeah, it changes the actual amino acids, which changes the physical shape of the proteins, which fundamentally changes how the virus actually behaves.
And this is where the math really comes in. By comparing the rate of these character rewrites, the non-synonymous mutations, which we call DN, to the rate of the harmless typos, the synonymous mutations, or DS, we get an incredibly powerful metric.
The famous omega ratio.
Yes, the omega ratio, or DNDS.
So omega is basically the golden key to reading the diary.
It really is. Let's break down the rules for it. If the omega ratio is less than 1, it means the rate of structural changes is lower than the rate of harmless typos. We call this purifying selection.
Purifying selection, meaning the virus is essentially weeding out the weak mutations to keep itself pure and functional. So extending our analogy, that's like a really strict editor immediately deleting any wild experimental changes to the plot because, well, the original story is already a bestseller. The virus is perfectly adapted to its environment, so any change is basically punished.
Spot on. That's a great way to visualize it. But what if omega is greater than 1? That is what we call positive diversifying selection.
So the editor is actively encouraging new, successful ideas.
Right. The virus finds itself in a new, hostile environment, like a totally new host species, and structural changes are being heavily rewarded because they help the virus survive the new immune system.
Okay, and what if omega is exactly 1?
If it's exactly 1, the evolution is neutral. The editor has basically left the building entirely, and changes are just accumulating totally at random.
Okay, got it. So the researchers in this Cell paper took this omega ratio, and they ran it through a highly advanced mathematical framework called the ReLAX model. And ReLAX measures a specific parameter called K.
Yes, the K parameter is crucial here.
And we need a good way to picture this one, too. Let's stick with our strict editor. Think of K as the amount of coffee the editor is drinking.
I like that. That works perfectly.
Right. So if K drops below 1, the editor is decaffeinated. They are asleep at the desk. Selection is relaxed, and mutations just kind of slip by. But if K is above 1, the editor just slammed 3 espressos. Selection is intensified. They are frantically rewriting and optimizing the code to deal with a new problem.
And the researchers were looking for this K parameter to spike in a very specific place on the family tree, what we call the stem branch.
Okay, wait, I have to stop you there. Because when I was reading this part of the source material, I was really scratching my head. How on earth are they measuring mutations on a stem branch from an event that happened years ago?
It sounds like science fiction, I know.
It does, because picture the family tree, right? You have a thick cluster of branches on the left representing viruses circulating happily in bats. And on the right, you have an explosion of branches representing the human pandemic. The stem branch is that solitary bridge connecting the two. But it's not like we had a Q-tip in that exact bat at the exact moment it coughed on a human. Are scientists just guessing what the RNA looked like?
It's a really great quote, and no, it's definitely not a guess. It relies on a super computationally heavy technique called ancestral state reconstruction.
Ancestral state reconstruction.
Okay, think of it kind of like linguistics. If you want to know what a dead language like Proto-Indo-European sounded like, you look at modern French, Spanish, and Italian. If they all share a similar word root, you can mathematically deduce that their common ancestor used that root.
Oh, wow.
Computers do the exact same thing with viral RNA. They take all the modern human sequences and all the modern bat sequences, and they mathematically work backward, step by step, to reconstruct the exact genetic sequence of the ghost virus that crossed the bridge.
That is absolutely mind-blowing. So we reconstruct the ghost sequence, and we look at the stem branch, and if the Hollywood framework is correct, and viruses must adapt to humans to cause a pandemic, that adaptation has to happen right there on that bridge.
Exactly. We should see the editor chugging espresso.
The K parameter should be through the roof.
Correct. You would absolutely expect to see a clear, undeniable signal of intense directional selection. So the researchers took this entire phylogenetic toolkit and applied it to the most devastating recent outbreaks.
Okay, so let's do a quick recap and a bullet summary for you guys listening so far, because we just covered some dense mechanics.
Good idea.
Bullet 1, we were reading the virus's RNA diary, looking for rewrites, not just typos. Bullet 2.
Yep.
We are measuring the genetic editor's coffee intake, the K parameter on the bridge between animals and humans. So put yourself in the shoes of a public health official for a second. If you believe a virus has to undergo massive, complex changes to survive in a human body, You're going to build your whole global warning system around looking for those changes, right? You'd expect the evolutionary gears to be throwing off sparks right before a pandemic hits us.
Which is exactly why the results of this study are so paradigm shifting.
Okay, hit us with the results.
So the researchers pointed their magnifying glass at the 2013 to 2016 West African Ebola epidemic, which was an event that devastated multiple countries. They looked at the 2004 Angola and Marburg virus outbreak, which had an incredibly high fatality rate. They looked at the 2009 H1N1 swine flu, Mpox, and of course they looked deeply at SARS-CoV-2.
The big ones. And did the editor wake up? Did the gears shift on the stem branch?
They did not. The results were shocking.
Wait, really?
Across the board, for all of these major natural spillover events, there was absolutely no detectable change in the intensity of natural selection on the stem branch. The K parameter stayed right around 1.
Meaning what exactly? Break that down.
Meaning the selection intensity was mathematically indistinguishable from the background reservoir.
Okay, hold on. I'm going to push back here. Because for years, the prevailing media narrative was that SARS-CoV-2 must have circulated in pangolins or raccoon dogs in a wet market, slowly adapting its spike protein to perfectly fit human receptors. Like it was the prevailing theory everywhere. Are these researchers saying that entire narrative is physically not visible in the RNA?
This raises such an important question about how we interpret early pandemic data. And the study addresses this. The researchers did a massive comparison. They took the earliest known human sequences of SARS-CoV-2 from Wuhan, and they compared them to closely related bat coronaviruses like RAT-G13.
Okay.
When they ran the ReLX model on the stem branch, separating them, the evidence of a prolonged adaptation period in an intermediate host simply wasn't there.
So it didn't spend months learning to infect us in a raccoon dog.
The data suggests it did not. The evolution of SARS-CoV-2 right before it exploded in humans looked exactly like its normal, everyday evolution while sitting in a bat. It did not undergo a specialized training montage. Now, to be clear, it may have physically passed through an intermediate animal, like a raccoon dog, but it didn't spend enough time there to significantly alter its evolutionary trajectory.
I see.
The selective sieve happens at the physical species barrier, the actual moment of exposure. These pathogens didn't adapt to become zoonotic. They were already capable of sustained human-to-human transmission.
Okay, here's where it gets really interesting to me. So it's not a training montage, it's the lottery.
Yes, the lottery.
Think about it. Millions of viruses are circulating in nature right now. Most of them hit the human barrier. Maybe a hunter gets a drop of blood on a cut, or, someone breathes in guano dust in a cave and the virus just bounces off. It can't replicate. But a few of them, entirely by chance, already hold the exact winning combination of amino acids to latch onto our cells.
Yes, they just need the physical opportunity to buy the ticket. And what's fascinating here is that while they don't adapt before the jump, they absolutely adapt after the jump.
Okay, so the frantic rewrites happen after the book is published in a new language.
Correct. The ReLX model shows this beautifully. Once the human outbreak is underway, the virus realizes it's in a vast new environment with totally new immune pressures, and the evolutionary gears do start turning.
So that's the variance we hear about.
Exactly. For example, during the COVID-19 pandemic, after SARS-CoV-2 crossed into humans, we saw it adapt heavily. We saw mutations arise that improved its binding affinity to the human ACE2 receptor. We saw the alpha, delta, and Omicron variants.
And we saw the same thing with Ebola, right? I remember reading that in the source text.
Yes. During the West African epidemic, Ebola evolved specific mutations, like the GPA82V mutation.
Right.
And that mutation increased its ability to infect human cells. But crucially, that happened during human-to-human transmission, well into the outbreak, not prior to it on the stem branch.
Okay, so making sure we have this locked in with another quick mini recap. For major world-stopping viruses like Ebola, Marburg, and SARS-CoV-2, that genetic editor didn't suddenly wake up and start making wild changes right before it hit humans. It was just business as usual.
Business as usual.
They hit the ground running, already fully capable of causing an outbreak, and all that rapid adaptation and mutation we hear about on the news, the scary new variants, that is a post-spillover phenomenon.
Exactly.
So a question for you guys listening. Does that change how you look at a forest? Knowing that right now, deep in a bat cave in Southeast Asia or a dense jungle in Central Africa, there are viruses harboring the exact genetic code needed to start a global pandemic, requiring absolutely zero evolutionary upgrades to do so.
It is a profoundly sobering reality. But, as scientists, whenever we find a broad rule like no pre-adaptation required for spillover, we immediately have to test the exception.
Usually you start to break your own rule.
Exactly. If no adaptation is the norm for a natural, direct spillover, how do we know this relaxed model actually works at all? What does it look like when a virus does spend significant time in a weird intermediate environment, or, say, in a laboratory?
Yeah, we need a positive control. We need to prove this mathematical tool can actually catch the thief when the thief is genuinely in the house. And the study uses the perfect test case for this: the original SARS outbreak from 2002 to 2004.
They did. And unlike SARS-CoV-2, the original SARS-CoV did show a massive, undeniable shift in its selection regime on the stem branch. The K parameter dropped significantly, down to 0.65.
Meaning the coffee wore off, the editor fell completely asleep.
Yes, this indicates a significant relaxation of natural selection before it achieved sustained human transmission.
But why the difference? Why did the original SARS leave this giant fingerprint on the evolutionary bridge, but the new one didn't?
Because of where it spent its time. SARS-CoV spent months, possibly even years, actively circulating in palm civets.
Right, the intermediate host.
Yes, before jumping to humans. That prolonged intermediate state in a completely new animal changed the evolutionary pressures on the virus. The rules of survival shifted. The RELOC-X model easily caught that distinct fingerprint. It tells us that if SARS-CoV-2 had spent significant time stewing in an intermediate host, it would have left an evolutionary trace. But it didn't.
Which brings us to the spiciest part of this cell paper: artificial environments. What happens when humans take viruses out of nature and put them in a lab?
Well, in a laboratory cell culture, a virus is essentially pampered. It is placed in a pristine petri dish with perfect nutrients, ideal temperatures, and most importantly, absolutely no immune system actively trying to kill it.
It's basically an all-inclusive resort for pathogens.
That's a great way to put it. And because it's at a resort, the evolutionary pressures vanish. The strict editor is gone entirely. So the researchers tested their model on viruses known definitively to be passaged in labs.
Okay, like what?
They looked at the live attenuated measles vaccine, which was passaged repeatedly through chick embryos. They also looked at a strain of H5N1 avian flu that researchers deliberately passaged between ferrets to study how airborne transmissibility works.
Let me guess. The K parameter caught them red-handed.
It caught them easily. The model showed extreme shifts in selection intensity on the stem branches of these viruses. In the cell cultures, they showed intense relaxation of selection because all the purifying pressure of a host immune system was suddenly removed. And in the ferret gain-of-function experiment, it detected the distinct, unmistakable signature of artificial selection.
This is just wild to me. You can literally read the RNA and tell if a virus was chilling in a natural bat, struggling to adapt in a civet cat, or just sipping margaritas in a petri dish.
Exactly.
Which brings us to a decades-old medical mystery that this paper actually helped solve: the 1977 H1N1 flu.
Yes, this is a fascinating historical case study. So in 1977, an H1N1 influenza virus emerged and caused a global epidemic, mostly affecting younger people. But when scientists finally sequenced its genome years later, they realized something impossible.
Okay, what was it?
The 1977 strain was genetically almost identical to strains that had circulated back in the 1950s.
It was a ghost. I mean, it's as if the virus just stopped evolving for 20 years. Right. And in nature, the flu mutates constantly. It doesn't just hit the pause button.
Right. So the prevailing theory in the scientific community has long been that this virus was accidentally released from a laboratory freezer where it had been stored since the 50s, or perhaps it was part of an aborted live vaccine trial.
Okay.
So the authors of this 2026 paper ran the 1977 flu through the RELX model to see what the stem branch looked like.
And what did the diary say?
It found a significant shift in selection intensity on that stem branch that perfectly matched the signature of laboratory passage. The evolutionary fingerprint of the 1977 flu was undeniably artificial.
That is incredible. We are basically talking about phylogenetics acting as a cold case forensic tool here.
It really is.
So just to gather our thoughts here for a final recap, this mathematical model successfully detects adaptation when a virus stews in an intermediate host, like the palm civets with the original SARS. It can easily spot the pampered evolutionary signature of a virus grown in a laboratory cell culture, and it confirmed the long-held suspicion that the 1977 H1N1 flu was a product of laboratory passage, not natural evolution.
Right.
Think about the sheer power of this tool, you guys. If a brand new unknown virus emerges tomorrow, how quickly could we run its sequence through this genetic fingerprinting to figure out its origin story? Did it come directly from a bat? Did it spend a year in a farm animal? Or did it leak from a lab?
The answer is very quickly. With modern sequencing, we could have that answer in a matter of days.
Wow.
And that rapid assessment is absolutely crucial for how we respond on a global level. Which really brings us to the ultimate takeaways of this study.
So what does this all mean for us? We started with the Hollywood movie plot, the virus mutating in the cave, training for its big moment. But the data says zoonotic pandemics are a lottery, not a training montage. Humans are constantly bumping into animal viruses,
Absolutely.
and the vast majority of them fail completely. The ones that cause pandemics aren't the ones that work the hardest to adapt to us. They're simply the statistical anomalies that we're already holding a winning ticket.
If we connect this to the bigger picture, it requires a massive structural paradigm shift in public health.
Yo, so?
Well, the traditional framework led us to believe we would have a warning period. We assumed a virus would enter a bridging host, like a pig on a farm or a civet in a market, and we could watch it warm up. We assumed we could monitor the genetic changes, see the editor waking up, and intervene before it fully adapted to humans.
But this study says the warm-up period doesn't even exist for most major outbreaks. It goes straight from the bat or the rodent right into a global crisis.
Exactly. This means our global surveillance strategies need to be completely reevaluated. We cannot afford to wait for stepping stone mutations to appear in intermediate hosts.
Right.
By the time we see them, it is already too late. The spillover has happened.
Right.
Our surveillance needs to be much broader, focusing directly on the natural reservoirs themselves. We need to be mapping the viral diversity in bat caves, in rodent populations, in primate habitats to understand what is already out there holding a winning lottery ticket.
We have to stop looking for adaptation and start looking for exposure. Where are humans physically intersecting with these reservoirs?
Precisely. The risk isn't that the virus will magically learn how to infect us. The risk is that we will simply give the already capable virus the physical opportunity it needs to cross over.
Which leaves you with something major to chew on today. If the viruses that will cause the next 3 or 4 global pandemics are already fully capable of doing so right now, sitting quietly in a forest canopy or a dark cave, not needing a single new mutation to wipe out millions, what does that mean for how we treat the planet?
That's the real question.
As climate change alters ecosystems and as rampant deforestation forces human settlements and natural reservoirs like bat colonies into the exact same geographical spaces, we are essentially buying millions more of those lottery tickets every single day. The boundaries are disappearing. Are we pointing our alarms at the wrong things? We spend billions analyzing how viruses might change, but maybe we need to focus entirely on how we are changing the boundaries between us and them.
It is a question of prioritizing our defenses. The enemy isn't training, it's just waiting.
The Hollywood script is wrong. The monster isn't leveling up in the shadows. It is already fully grown, and it's just waiting for someone to open the door.
Thank you for joining us on this deep dive into the source material. We hope this gave you a whole new lens to view the natural world and the microscopic threats within it. Keep asking questions, keep challenging the narratives, and we'll catch you on the next deep dive.